those in our immediate environment, those within our social context, when they observe our pain expressions, when they observe us going through pain, they appraise and they observe these behaviors, and then they choose to engage with you, potentially help you, and that engagement and whether or not they do offer help or they don't can alter your own pain perception and can alter the expression of pain. And so that's really at the crux of what we study in the lab, is at least half of the lab studies the social influences on pain. But as I mentioned at the top of the lecture, we're not the only, well, we study primarily mice, and mice are an extremely social species, as are many other species, including rats. But I want to take a step back a little bit, because the first thing that I'm going to talk to you about is some work that we've been doing in the domain of empathy, in understanding the mechanisms that underlie empathy. And so we use mice in our research, and so we don't study the traditional concept of cognitive empathy. And when you think of empathy, you may think of placing yourself in someone else's shoes. Mice, you can't really do that. But empathy is not really one thing. It's actually a constellation of behaviors. And so it can be something very simple, such as emotional contagion behavior. And emotional contagion is if one baby cries, all the babies cry. And so think of the way hospitals used to be set up with nurseries, right? One baby cried, it usually elicited distress and crying in the other babies in the nursery. So that's an example of something like emotional contagion. Sympathy and compassion, pro-social behavior, perspective taking, empathy, these all relate to behaviors that are all related to, well, I guess empathy. And they can be sort of divided into primary, secondary, and tertiary. And as you go up that staircase, the behaviors become more and more complex, right? But on each level of the staircase, different animal species have been shown to possess those capabilities. And so we study primarily mice. I believe this image right here is the mouse. And so we are kind of at this bottom step where we're studying emotional contagion, at least to begin with. And so just to harp on that point a little bit, is that emotional states can be contagious. And so I am here to talk about pain, but think about sadness, joy, fear, disgust, and anger. A lot of these emotions can be very powerful, and being around sad and depressed people can also make you feel a little blue. Being around happy, joyful, positive people can actually evoke some of the same feelings. The same thing for fear, disgust, and even anger. Anyone who has gone through, having small children can easily relate to how anger can be contagious. Especially, I know that was the case for us. But we've been asking the question, well, can pain be contagious, right? And here, pain, and I think this is kind of an important point, is that pain we typically think of as a sensory experience, but it's also an emotional experience, right? When a young child hurts themselves, what do? They cry, right? Crying is, at the heart of it, an emotion or an expression of an emotion. And so to study something like emotional contagion, which is a very primitive form of empathy, we developed this mouse model in which we use a very old pain test. And so these are mice in which we take a single mouse and then we inject that mouse in the belly with a very dilute amount of acetic acid. And what that does is it causes a little bit of gastrointestinal distress, kind of like we give the mouse a little bit of a tummy ache. And we do this because it evokes very reliable and very measurable pain behaviors. And so when an animal is injected with this pain stimulus, they elongate their torso and they constrict their abdomen. And we can count up the number of times the animal does this as an indication of the degree of pain they are experiencing. And so when we do this, we have three primary conditions. We have a condition where a mouse is tested alone. So this gives us an idea of, well, how much pain is caused by the stimulus. And then we manipulate the social context. We have a condition where we have two mice now in the same testing arena. One is injected with acetic acid, one isn't. And then we have another condition where we have two mice injected with this pain stimulus, right? And this becomes the critical condition for us because now we're asking the question, how does the pain of one mouse influence the pain of the other mouse? And so when you look at it, this is the level of pain behavior that is normally evoked by our pain stimulus. When you only have one mouse in pain, but with a social partner, the level of pain does not change too much. But now, when you put two mice together and they're both in pain, you have this amplification, you have this facilitation of pain. But the punchline here is that it's only true for cage mates. It's only true when the mice actually know each other and are familiar to one another. If they're strangers and we take them from different cages, we actually don't have this response. And so we've spent quite some time looking at, well, what's actually preventing pain contagion in strangers? Because we view contagion and pain contagion as a form of empathy. And so if we can sort of figure out why strangers don't actually engage in this, we might be actually getting at some of the mechanisms that block empathy. And so a lot of this work was done by a former PhD student in the lab. And she had the hypothesis that, well, strangers, when they interact, there's a little bit of uncertainty, right? There's a little bit of, you don't know really this individual. You're a little wary of them. And so she had the idea that, well, maybe it's stress. Maybe stress is actually blocking pain contagion. And so what she did is she used this drug called metirapone. Metirapone blocks the primary stress hormone. It blocks cortisol or corticosterone in mice. And when she gives metirapone to the strangers, she now evoked this pain contagion. So on some level, stress is actually blocking and preventing pain contagion. Now I'm going to show you a lot of data. And I'm going to do my best to interpret it for you and sort of give you the take-home points. But she spent a lot of time actually looking at and searching for the brain regions that were important and that were underlying this. And she looked at stress receptors and the activation of stress receptors across a number of brain regions in the mouse. And she honed in on this one brain region right in the front of the brain. It's called the medial prefrontal cortex. And it's very important for pain processing. It's also very important for social cognition, distinguishing between others and yourself. And what she found was quite a bit of stress receptors activated in this region. And so she took some mice and she went in and she injected into the mice, directly into this area, a drug to specifically block these stress receptors. And so she injected this drug called RU486 right into this medial prefrontal cortex. And when she does that, now she also elicits this pain contagion. When we inject this drug into other brain regions, we don't get this effect. And so there's something about stress receptors being activated in this front part of the brain that's actually preventing pain contagion. And you can reveal it by alleviating some of that stress activation. Work that I did when I was a postdoc is we asked the simple question, well, what about pain contagion in people? Right? And so it's especially something like pain contagion, how relevant is it across species? And so we we brought in, and this was when I was at McGill, we brought in a number of undergraduates into the lab, and we had them undergo this really, really old pain test. It's called the cold presser test. And so it's exactly like this image shows. It's a very cold water bath, and the subjects place their hand in this cold water bath, and they rate their pain after keeping it in for about 30 seconds. And we had them go through a number of different conditions. And we ran 3 different studies, and now I affectionately refer to this series of studies as Friends, Drugs, and Rock and Roll. And so I'll walk you through these studies. This first one here is the Friends study, in which we had, and we replicated that initial mouse study, in which we have friends and we have strangers. And we bring them into the lab, we test them for their cold pain threshold, either in the presence of a friend or in the presence of a stranger. When friends are tested together, they rate their pain as more intense and more unpleasant, right? So their pain intensity is actually higher. Lucky for us, that same drug that we gave to the mice, metirapone, is also clinically approved for the treatment of a few clinical conditions. And so we can give that to people. And metirapone, if you just have strangers come through the lab and you give people metirapone before meeting their friend or stranger, you can actually elicit this pain contagion. And so even in people, if you block the stress response, you can elicit pain contagion. And then the last thing, and this is what we couldn't do in mice, is we can't really give mice a social bonding experience, but we can do that in people. And so what we did is we had people come into the lab, and they were all strangers in this experiment, and they played a couple of rounds of the video game Rock Band together. And so Rock Band, if you're not familiar with it, you have these plastic guitars, and there's notes that fly on the screen, and you have to press the keys that correspond to the notes. And after people had played Rock Band together a few rounds, and you test them for their pain responses, together, they're actually reporting more intense pain. And so we've done more work in that area on pain contagion, but I had a PhD student, and she's just finishing now. But a number of years ago, when she first started, she said, okay, the pain contagion work is great, but what about more of an empathy-like model, where you have an observer that is observing a pain response in another? How does that change the pain responses of the observer? And so she came up with this really neat paradigm, and this is all work from Sandra Pulse. And so the model is there's a social interaction between an observer and a cage mate, right? So we have 2 mice, they're socially interacting together, and then she takes one of those mice and she injects that mouse with a pain stimulus. And then she pairs them back together, right? But unlike our contagion experiments, what Sandra did is she tested the observer. She had tested the observer for mechanical sensitivity. And so what she finds is that in mice that just socially interact, there's no change in pain sensitivity. But if you have a mouse that interacts with a cage mate in pain, now you have this increase in pain sensitivity that lasts about 4 hours. It resolves by about 24 hours. But since we're interested in the neural mechanisms and the neurochemicals behind a lot of these responses, we asked ourselves, well, what could be involved potentially in mediating this enhanced pain sensitivity? And what Sandra hypothesized is that there's a specific chemical called cholecystokinin, or I'm going to refer to it as CCK. And CCK is released when there's a threat to safety. And so you can think of that situation where there's an animal in pain and an animal not in pain. The animal that's not in pain, there's a little bit of a threat to that animal because now it doesn't know what's happening with its cage mate, even though that cage mate is now experiencing and expressing pain behaviors. And so CCK is released when there's a threat to safety. CCK results in panic responses, and CCK also enhances pain responses. And so the first thing that Sandra did was she used this drug that blocks the CCK receptor. And so if you look at the interface between neurons, right, one neuron releases the chemical that binds to receptors on another neuron, and Sandra gave this chemical, this drug called proglumide, that actually blocks CCK receptors. And so it prevents CCK from doing its job. And when she did this, when she pre-treated mice with proglumide, she completely prevents the enhanced pain sensitivity. So it seems like there's enhanced CCK release that's causing and creating this enhanced pain sensitivity. And so you might not be able to see my nice fluorescent images here, but Sandra spent a lot of time looking at, well, which brain regions are activated. And so there is some, some nice red dots on some of these images. They're a little bit faint to see because of the light. But those red dots indicate that a neuron is active. And what she did is she counted up all the red dots. She counted up all the red dots, and she discovered that there's one specific region of within the middle of the brain, and it's called the periaqueductal gray. And the periaqueductal gray is very important and acts as a pain hub. And so pain information that is coming up from the spinal cord or coming down from the brain goes through the periaqueductal gray. And so a specific region of this brain structure, the lateral PHE, there is much more cells that are being activated, and you have neurons that are being activated in those regions in that condition in which you're observing, or you have a mouse observing another in pain. So in her first experiment, she injected proglumide just into the, just systemically, and so proglumide is going to go everywhere and block all CCK receptors. But she approached it from the perspective of, if this brain structure that she's identified is important, and CCK receptors in this structure are important, then if we inject specifically this drug proglumide into the lateral PAG, we should actually block this enhanced pain sensitivity. And so these are our control mice, and so you can see following the social interaction, there is more pain, there's enhanced pain sensitivity. But when we micro-infuse proglumide directly into this brain structure, we don't have the enhanced pain sensitivity. And so within the periaqueductal gray, the lateral division, we have enhanced neuronal activity, and that enhanced neuronal activity seems to be driven by the activation of CCK receptors. And so there are many different brain regions that actually send signals to the periaqueductal gray. And one of the things that Sandra wanted to do is identify, well, are cholecystokinin neurons coming from another region which is activating this brain structure? And so what she, and all of these regions are important for things like empathy, social cognition, and even pain processing. And so one of the things that Sandra did is she used a combination of a transgenic mouse and a viral technology. And so what she did is she took a transgenic mouse and she injected a virus into the lateral PAG, and that virus is going to infect only neurons that contain cholecystokinin. And that virus is then going to be transported and shuttled back to any brain structure that is actually projecting into that region. And what she identified, and this is the first time that it's been identified, is long-range cholecystokinin neurons in the anterior cingulate cortex that project to the lateral PAG. And so the anterior cingulate cortex is this brain region that's been important that's really important for pain processing and for social cognition. And so here we have a brain region that is important for empathy and social cognition and pain processing that's projecting to the lateral PAG that contains specifically cholecystokinin neurons. And so to understand whether or not these neurons are actually the neurons that are responsible for the enhanced pain sensitivity, what she did is she used a technique called optogenetics. And in optogenetics, and in her approach, she also uses a combination of a transgenic mouse and viral technology. And now here, what she's doing is she's injecting a virus into the anterior cingulate cortex. That virus is going to be expressed only in cholecystokinin neurons. But that virus is then going to be transported to any of those regions in which they project to. And so for us and for our purposes, the lateral PAG. But what this virus is doing is, yes, it's infecting the cholecystokinin neuron, but it's also allowing the neurons to now express a light-sensitive protein. So the virus is actually carrying a light-sensitive protein that we can then implant a fiber into the mouse to shine light specifically onto different neurons to either turn the neuron on or turn the neuron off. And so for Sandra's purposes, what she wanted to do is express these light-sensitive proteins and turn off, specifically shut off, the cholecystokinin neurons. And so in our control condition, right, when we inject a control virus, we inject into the lateral, or into the anterior cingulate cortex, and then we shine light into the lateral PAG, but it's a control virus. It's not expressing this light-sensitive proteins. The animal still goes through the normal pain response, and there's pain expression. However, if we inject and we express a light-sensitive protein, and we shine light in the lateral PAG, and we turn off specifically those cholecystokinin neurons, we now don't have this enhanced pain sensitivity, right? And so what this is showing is it's actually showing for the first time the circuitry in which that may subserve and underlie empathy and the enhanced pain sensitivity that we experience when we observe a loved one going through pain. But it's not all bad. So even though I've focused on enhanced pain sensitivity, right, social interactions, especially positive social interactions, can actually reduce pain, right? And so it goes both ways. And so this was work by Nabi with her when she was in the lab, and she came up with this this really neat protocol. It's called social reunion analgesia, and this is what we've termed it. And so when mice are born, they're born in litters of 6 or 7, and then they live with mom for about 3 weeks. After the 3-week time point, they're then separated, and then they're then paired up into separate cages, right? And so now they're old enough to get their own cages. But in adulthood, we take separated siblings and we reunite them, right? And so these mice live together for the first 3 weeks of life, and then they haven't seen each other for 5, 6, 7 weeks, and then they're reunited, and then we test their pain sensitivity. And so what we find is that sibling mice who go through this reunion, they actually have decreased pain sensitivity. Stranger mice interacting, they don't, and mice tested alone, they also don't have changes in pain sensitivity. But the really neat thing with this is that it's only true for females. Female mice are the ones that show this lack of pain sensitivity or this social reunion analgesia. Male mice don't seem to care. And so, to summarize a few years of work into one slide, what Nabi did is she looked at, again, a few chemicals and brain regions that might be activated. And what she honed in on were oxytocin neurons in a specific part of the brain. And so oxytocin is notorious for being dubbed the love hormone. It's important for social bonding. And in female mice only, not the male mice, this is only the female mice, mouse data. In the female mice, they have activation of oxytocin neurons. Male mice don't, right? And so we think that it's these positive social interactions in adulthood from the females are actually causing activation of oxytocin neurons, which is leading to this enhanced analgesia. So you can't really see this one all that well. This is, again, some work from Navi, and she also focused on the PAG, and she counted up, again, all the red dots, and she finds that a different area of the PAG is activated. But this area of the PAG seems to be more important for pain suppression rather than pain facilitation. And the final piece of data that I'm going to show you is data from the PAG in which we use a transgenic mouse. And that transgenic mouse has a fluorescent protein embedded in the gene that codes for the mu-opioid receptor, right? So the mu-opioid receptor is the primary target of things like morphine, endorphins, right? And so it's very well known in the opiate field. And what we find is that in the ventrolateral PAG, we have neurons that are activated that contain the mu-opioid receptor, and they are in quite close proximity to these oxytocin fibers. And so what we're working on now is we're working on understanding whether or not oxytocin is actually interacting with the opiate system to create social reunion analgesia. And we do believe that the opiate system is important for social reunion analgesia because when we block the opiate system by giving a drug called naloxone, we can actually reduce and eliminate social reunion analgesia in female mice. So, some of my take-home points. So pain is going to be our most protective device, and it's changed by our experience in our social context. Understanding pain sensitization and the brain mechanisms of pain modulation may help with the identification of new pain therapies. It also, especially as far as social modulation of pain goes, it also removes a little bit of the stigma associated with social factors that actually modulate pain. And social behavior can change pain perception to enhance or reduce pain, and this depends on the nature of the social interactions. And so whether or not the social interaction and what the mice, in our case, are experiencing, if it's viewed as threatening, it may actually enhance pain. But if it's a positive, soothing, calming experience, like as in the social reunion analgesia, which we view it as a positive experience, it may actually work to reduce pain. And so I'm just going to thank people in my lab. This is a little bit of an older picture. Some people are no longer in the lab. We do have some new additions, but most of the work, well, actually all the work that I talked about today was done by Navi and Sandra, our two PhD students in the lab. And so I'd be happy to answer any questions or further interpret any of the data that you might be curious about. Thank you.
As a neuroscientist, this is his life work, and those other researchers who worked with him. But he's had to distill this so that we can understand it. Do we have questions? Yes.
Just curious in terms of, you talked about in the human model with pain projection. How did you determine when someone was exhibiting pain, or how does the counterpart exhibit it? Was it, I've seen some studies suggesting that swearing, etc., during pain can help alleviate or prolong some of the pain experience or reduce the feeling. So I'm just curious how you.
Yeah, it can, and so swearing, for instance, as you bring up, can also be a psychological, a cognitive thing that can actually work to reduce pain. In the study specifically with people, where we're using the cold pressor task. So all participants place their hand in the water for about 30 seconds. And then after the 30 seconds, so it's pretty cold water, it's like about 2-3 degrees Celsius. And then after the 30 seconds, they remove their hand and then they rate their pain, right? And so they rate their pain on a scale of 1 to 10. And we decided to go with that rather than have them hold their hand in as long as they can, because we didn't want the behavior of one participant to influence the behavior of the other. And so we had them rate their pain sort of anonymously.
Any other questions? Yeah, in the back there.
It possibly could. And so for most of this project and what we've done is we've kept it relatively the same. But this was based off of an older study published in the 90s in which they used a longer time frame. And so I'm willing to bet that even if we extended it out, so we usually test mice at 8 to 9 weeks, if we extended it out to 11, 12, maybe even 13 weeks, we'd still find very similar results.
Just curious, in terms of potential practical applications, I know that this is obviously in mice, but any potential advances in terms of pain care instead of relying solely on opiates or opioids? Maybe, you know, if you're talking about sort of a positive environment, any sort of potential research that you're aware of that looked at that instead of solely relying on?
So I think like a lot of what this does, right, because like when you look at pain, especially the development of chronic pain, and I didn't talk at all about chronic pain, but chronic pain actually I have it on the next slide, actually operates in a biopsychosocial sphere, right, where you have biological, psychological, and social influences all impacting. And so what a lot of what we do sort of gives, reduces the stigma associated with, well, maybe, you know, social interactions are not so meaningful. Maybe they should be, you know, more or less dismissed in terms of patient care. And but when it comes down to it, right, providing actual biological evidence that some of these either psychological manipulations or social influences actually do change biological processing and biological circuitry actually reduces a lot of the stigma that chronic pain patients often do face, right? And so when you look at individual patients, right, the influences on that individual patient, right, like the biological influences, the social, and even the psychological influences can have different bearing on each patient, right? And so it really has to be, and I think the big thing in the pain field right now is personalized pain medicine, right, where you have to take an individualistic approach and really fully understand what's going on with that patient. And that's potentially where cognitive behavioral therapy and different interventions like that will probably come into play.
And yes.
So I have a question about different species, like birds, for example, especially live in sloths, and birds tend to hide their symptoms for survival reasons. Did you notice anything similar? And
So
sometimes tend to hide their symptoms just to appear more stronger than they are.
yeah, so I do believe on some level that that we do, right? And so there are, I haven't done any of this research, but depending on, like, even if you just take our species, humans, right? Depending on the sex of the participant and the sex of the experimenter, right, that can actually change the expression of pain, right? And so if you have a male participant and a female experimenter, the male participant may want to appear like the pain stimulus actually doesn't bother them, right? It's kind of like the Macho Man effect. We published a study a number of years ago actually showing that mice hide their pain expressions when there's an experimenter present. And so most of these experiments are actually done when there is no experimenter present and we videotape, right? But if you, but in that study, it was actually only the men who caused the mice to hide their pain expressions. The females didn't really have much of an effect.
Wow.
Yeah, and it seems to stem from axial secretions, right? And so I remember a lot of the press coverage on that study at the time said reeking male scientists reduce pain expressions in mice. But yeah, and so mice, for instance, they're prey species. And so it makes sense from an adaptive perspective that they're going to want to hide their pain, because then it just makes them more vulnerable. Yeah, good question.
Yeah.
So I have two questions. First of all, just for clarity, for my sake, so does the pain lab only focus on physical pain? Do you do anything around emotional pain?
So we only focus on physical pain.
Okay, fair enough.
Yeah, because, well, especially studying social pain or emotional pain becomes really, really difficult in an animal model.
Okay. And then the other question that I have is, when you're looking at sort of the findings in human beings, wherever you've applied it, is it consistent across all age ranges? And because I deal with a lot of elderly people, and I'm often told, they don't feel pain or pain. And then the second part of that question is, has any work, whether in your lab or anywhere, been done with dementia patients?
So there is a researcher out at University of Regina who does a lot of work with dementia patients.
Okay.
In pain responses and pain perception. The part of the issue with dementia patients is the communication pain, right? Because they're not able to properly communicate that pain. And so because they're not able to properly communicate and rate the pain, we just naturally assume that, well, maybe there's, they're experiencing less pain, but that's not necessarily true. But there is also an age dependency associated with that, which could be driven by biological aging, or it could also be like a rating and a reporting issue as well, right? Because older individuals are more likely to have experienced many pains along the way in their life. And so, just because they wake up with a little bit of a sore back, or like they're getting old, and so of course they're going to hurt, and so they don't really report it as much. But there's not a lot of lab-based research on it to actually test pain sensitivity, as far as I'm aware.
Thank you.
Any other questions? I have some. Yes, I was going to go to the chat next, unless there's any live questions. One more here?
Just last one. In terms of the oxytocin potential and source and MU, kappa, et cetera, pain reduction model, I know with certain substances coming more into the foreplay that play on oxytocin and MDMA specifically, have you seen any sort of pain research done with that substance in particular, looking at the role that oxytocin plays?
Like with MDMA specifically?
Or anything that elicits a flooding of oxytocin. Or a practical application that could be, an adjunct therapy.
Yeah, I mean, I don't really know of anything that will cause like oxytocin release specifically, per se, and whether or not that has been looked like. MDMA and some of the psychedelics, they are being trialed and looked at for their effect on pain perception, for sure. Whether or not those reductions are mediated through an oxytocin specific system, harder to say, I think. But we do have evidence in the lab that oxytocin and something like morphine or an opiate could be used as adjuncts together, right? And so the benefit of that would be that you would have to use lower dose opiates while supplementing with oxytocin to achieve, more or less the same analgesic outcome.
Yeah, curious.
Yeah.
The current, great crisis across Canada and the world.
Yeah, I mean, it is kind of a big thing. Even the other drug that I was talking about, proglumide, there's older research using proglumide and focusing on the CCK system that actually can improve opiate-based medications and improve opiate outcomes. Yeah.
Any other questions from the floor? I have, yes.
So you had a slide about siblings, right? And then, they're more empathetic if they're female,
Mm-hmm.
right? Do you do the same study for strangers, female versus male, or combined? Is there any difference?
Yeah, we have, like, in that one slide, especially with the siblings where we reunite them, if that's what you're referring to, we did include strangers. And so we did include the female strangers, we included the male strangers, and so we had all of the different social relationships across both sexes. In most of our experiments, we do include both sexes. That was just the most striking and stark sex difference that we actually did find. But yeah, we do include both siblings and strangers.
Quite similar to siblings?
No, for the strangers, they don't have the like if you just have two strangers interacting together, there's no change in pain thresholds. It's just when you have those siblings and they're going through this social reunion, and we really view that social reunion as a positive experience, right? And we do think that on some level genetic relatedness is necessary for it.
I'm gonna ask you. Oh, yes.
Great presentation. Just a very curiosity-based question. I understand that you said that with the people, you ask them to rate their pain when they put their hand in the water. I'm just wondering, like, in the future, based on, I don't know the biology of it, but like how much the neuron lights up or how fast the signal goes or something, do you think we can potentially mechanically check the level of stress or the level of pain in each person?
Yeah, so I think what you're really referring to is whether or not we'll ever be able to find an objective measure of pain, right? And so that becomes really, really difficult because pain really by definition is a subjective experience, right? And so all of these things can change our perception and our experience. And so really honing in on this one, and even I started the talk by sort of outlining the pathway, right? And so there are very well-defined brain structures that are important for pain perception, but it becomes almost important or impossible to look at like this one brain structure and say, if we have activation here, we're definitely going to have pain, right? And so it becomes really, really difficult to actually have an objective measure, although people are still trying to develop one.
Yeah, I can think because one person's pain tolerance might be completely different than the other. So I'm wondering, like somebody who actively goes on like cold water swims or something might find it less hurtful than the one who's never.
Right, and so a lot of that, especially going on cold water swims and all of these experiences, are driven by your expectation, right? I don't find that would not be pleasant to me, right? That would be excruciating, and so I would probably find that extremely painful. But other people find it quite pleasurable, right? Like even you take the athletes who go through cold baths, right? And so they do that with a very specific purpose in mind, and that's for recovery, and that's for, performance enhancement. But me going through a cold bath, that's just torture, right? And so, like, that also highlights the point, is that the context, the meaning, everything about that situation can really change your perception of it.
Any other questions on the floor? I'm going to read a virtual question. We're back to animals. Is it possible for different animals to feel and share pain of one animal and create solidarity and union together in combating a threatening force like a predator?
That's an interesting question. Short answer is I don't know. Because mostly, like, and I think that there are some anecdotes that speak to this, in which, and I'm sure there are anecdotes out there in which there are, you know, two different species that may sort of team up to combat a predator or, you know, help one another. But those cross-species studies are rarely done in the lab, and it's really hard to, you know, sort of get at those studies. But, you know, I mean, you could think of, you know, different species who live together, like even like cats and dogs who live together. You know, even though they're not typical companions, if they do live together, they may have, you know, formed some sort of bond, and so there could be, but really hard to say.
So a very interesting question, we'll never know.
Very, very interesting question that we'll never know.
Okay, and the other question is specifically about cats. Any studies done on cats related to social impacts on their pain, both between cats with other cats as well as between cats and humans?
Another interesting question.
Do you have a cat?
I don't have a beagle. He's very loud. So specifically to look at like pain responses in cats becomes difficult to do because they're companion animals. And so really, and you know, animal research, and I'm not saying that mouse and rat studies are not highly regulated, because they are, but there is an extra level of scrutiny to looking at, especially something like pain in companion animals. And so it becomes really, really difficult to actually fully address that, unless you are just looking at specific case studies. But then if you're looking at only specific case studies, you maybe have like an example of one or two.
Anecdotally, cats hide their pain.
And anecdotally, I'm sure they do. Something that I didn't talk about is that most animal species actually express their pain through their face too.
Really?
Same thing as with cats. Is they will. If you have an animal that undergoes surgery, and so if it's you know, you have a cat that's being neutered or spayed, and then for a period following the surgery, there actually will be a distinguishable pain face. Mice are no different, rats are no different, and you can quantify it. And we do quantify it in the lab. For mice, not cats.
Okay, now we're back with another human question. If women are able to tolerate pain better than men due to social expectations and stigmas, I've got to reword this. I was going to ask if women being able to tolerate pain better due to different social expectations and stigmas that affect biological processes.
On a whole, women are actually more sensitive to pain. Women primarily make up the majority of chronic pain patients. It's probably 70% female. And across any pain test that we run in the lab, and that anyone's really run in the lab, females are more sensitive to heat pain, mechanical pain, pressure pain, ischemic pain, chemical pain, you name it. Females are more sensitive.
Sensitive or tolerate? Sensitive.
Yeah. So, like, there is, I think, a really big misconception that females are actually able to better withstand pain, and they have a higher pain tolerance. It's actually not true. And I think that stems from probably the fact that women have to undergo childbirth, right? And so it's maybe naturally assumed that they have a higher pain tolerance, but it's actually not the case.
Are we learning things here? I don't have any other virtual questions. Are there any other questions on the floor? There's been a lot of interesting questions that people have been listening. I think with that, we'll end the session. Thank you very much, Professor Martin.
Okay, well, you're very welcome. And thank you.